Hello and welcome to this PIR live event webinar brought to you by Partners in Research Canada. My name is Stacey Joyce and I will be your host. Remember that you can ask questions anytime throughout the presentation or the question and answer period by clicking chat near the top left of your screen. Make sure you change the to fields to everyone instead of all panelists and include your name, school, or city, and we'll give you a shout out when we ask your question. Today's guest is Dr. Jane Heffernan. She is a York Research Chair in the Department of Mathematics and Statistics and the Director of the Center for Disease Modeling at York University. Thank you so much for joining us. I'll let you take it away.
Thank you for having me, firstly, and thank you to everyone for joining this live event. I'm very happy to speak to you about disease modeling. So what I'm going to do first is I'm just going to get the presentation up and going. Okay, and so what I'm going to talk to you today about is math, vampires, and the NHL. And you'd probably don't understand how the three could be related, or how math could be related to vampires or the NHL. And hopefully, after the presentation today, I'll be able to give you some knowledge about that relationship. So, firstly, before I start to talk about vampires and the NHL, I just wanted to give you an example of where math is found in nature. Okay, so these numbers that are at the top here, these are called the Fibonacci numbers. This is the Fibonacci sequence. And what this says is that if you start with the numbers 1 and 1, then 1 and 1 is equal to 2. And then 1 plus 2 is equal to 3. Then 2 plus 3 is equal to 5. And 5 plus 8 is equal to 13. And 3 plus 5 is equal to 8, and 5 plus 8 is 13, and so on. So you can see that the next number is just the two previous numbers being added up. And that's what this little equation is that's down on the left, but you don't need to worry about that. So what's important about this sequence is that it's found everywhere in nature. And I give you some examples of some things down here. So briefly, I'll just say that almost all flowers in nature have a Fibonacci number of petals, and almost all have a Fibonacci number of branches. And if you think that you've seen a flower that has 6 petals and you see that 6 is not on here, well, usually that means that they have 2 rows of 3 petals, and you can see that 2 and 3 are here. If you look at a pineapple, the number of spots it takes for you to get up from the bottom to the top on a pineapple, that's a Fibonacci number. And there are 3 ways to do that on a pineapple, which is also a Fibonacci number. And each one of those 3 ways has a Fibonacci number of spots. So I invite you to look at these numbers and go through nature and look at trees and bushes and flowers and whatever you can think of to see if you can find these numbers. And also an interesting thing about these numbers is that if you divide 1 by the 1 before it, there's this ratio that we get as long as the numbers are large enough. And that gives us the way that a lot of spirals are shown in nature as well. And so I give you a lot of these spiral examples. You can see from the beginning of in the center of the sunflower, it spirals all the way out. And there's some sort of plant on the right side, a snail shell. You'll be able to see that spiral. And then also on the bottom left, I'm showing you a hurricane. Well, that's not an actual picture of a hurricane. That is a computer generation of a hurricane that you would see in a movie. And what they use there is the Fibonacci golden ratio. And then they also use something called fractals to be able to generate something that looks like a hurricane so that in the movies, you see something and you think it looks like a hurricane. Same with mountains and things like that. Okay, so now I'm going to get on to some work that I do. So I'm an infectious disease modeler. And what I do is I look at how your immune system interacts with different diseases that you get. So like if you have a cold or if you have the flu or if you have measles, things like that, and hopefully you don't. I also look at how well vaccines are helping your immune system stop pathogens from getting into your body. And then what I do is I look at how those pathogens and your immune system interact at the population level so that I can look at the spread of how diseases spread at the population level. So if you have a cold and then your friends get a cold, well, that's something that I look at. And then you can think that in your class, maybe there are some people who are sick. And then in the school, there are people who are sick. And then your school is part of a community. Your community is part of some network hub in the general area. And then you probably have an airport nearby. And then so your network community is also connected to the rest of the world. And so we look at how diseases also propagate globally with all of these stages. So then we're also interested in looking at behavior and how maybe you stay home when you're sick, or maybe in some places you'll wear something over your mouth so that you can't transmit a disease. So we're just really interested in looking at all the different types of behaviors that people have that help them stop getting a disease. Okay, and so that's really what we look at. So we go from the cells, which are the bottom of the screen, to a small class, to a school, to a community in a school, and then over to maybe the world network. So those might be airplanes going in between different cities, countries. Okay, so what I use, what to do my work is really just based on compartmentalizing a population into infectious disease classes. And this is a simple example where I can take a population and say, these people are susceptible, so they live in that S box. And these people are infectious, and they're in the I box. And these people are recovered and immune, and they're in the R box. And then if I model them by shaking hands or hugging each other or just breathing on something that's close by someone else, then I can look at the progression of susceptibles becoming infected people recovering. And that gives me what this graph is. And so you can see here, the red line is the infected class, and it's going up, and then it's going away. So that might be something like the flu season. And that information can help me understand how many people we should have vaccinated in the population so that we can effectively protect the entire population from getting a disease. Okay, so something that we did for fun was we tried to apply a similar model to a zombie plague. And so what we did, so this is the process that I work in, is first I look at all the literature to see if I can find historical documents that can give me an idea of what the different compartments of the population need to be in the model, and then also what the parameters are, how fast the disease is transmitted, how fast people recover or die from a pathogen. And so what I did is my collaborator and I, we went to all of the Hollywood movies, and we tried to see how zombies have evolved over the years. Are they slow? Well, in some movies, they're very slow moving, but in more recent movies, they're very fast. And in some movies, they're not very smart, and in some movies, they're very smart. And so we tried to figure out what our parameter values would need to be and our compartments from this type of information. So I'm also giving you here like a spatial map of living dead outbreaks in Pittsburgh, so and it also involves spatial aspects. And then down at the bottom, this is a Google search term for the word zombie. And what we use in my field is Google search terms to figure out where the disease might be. And so you can see that someone in your hometown has searched the word zombie in this map. And so maybe there's a zombie outbreak where you live. So then I can use all of that information to figure out what my model should look like. And so I can have susceptible people. Susceptible people can become zombies. Zombies can die, but the dead can also be reanimated back to the zombie class. In some movies, there are some recoveries from the zombie disease, so we involve that. And recovered people can die, but then be reanimated. And then also you can think that if someone has recovered, maybe they can become susceptible again, because maybe it's like a cold and you can get a cold more than once. So then we involve all that information into the model, and you can see that we get extinction of the human race in about 1 month if there's a zombie plague. So that's kind of fun. And also, thank goodness zombies aren't real. So then we can think about control strategies, like vaccines. What's the outcome that we want to happen in a disease model? So some of these people that are now zombies were family members, so we don't want them to die. So we're going to try to find some nice coexistence of the two populations. And then we can also translate the disease modeling from zombies over to maybe the spread of an idea. And so this is something that 2 of my students worked on with me, is trying to determine where a good location is for NHL expansion. And what that really is based on is trying to understand how chronically infected fans can infect their friends with ideas, so that their friends become fans of a team. And because really a team needs fans so that the team can sell merchandise and make money. So we looked at all the different NHL teams, and we tried to get an idea of what a good locational quality is. And then, so here you can see in purple, those cities had good locational qualities, which is based on the size of the city, the amount of money the city has, and then also the average temperature over the year, and if the city experiences winter. And so you can see the purple cities had good locational quality and the yellow cities didn't. And then the red stars are the 5 cities that we looked at: Seattle, Las Vegas, Kansas City, somewhere in the GTA, and Quebec City. And you can see Quebec City got a purple locational quality, which is good. Las Vegas got a yellow one. So then we looked at how successful teams are, because the successful team has more fans. And the purple teams are the very successful teams over the past 10 years, and the yellow teams are not. Okay, you can see that I'm in Toronto. Toronto doesn't have a very successful team. However, we have a lot of chronically infected fans. Then we tried to involve all of that into a spread of an idea model. And what we found was that in order for a new expansion team to be very successful in getting chronically infected fans, they need to have a success rate that's as good as the Boston Bruins have had in the last 10 years. But usually a brand new team has a success rate similar to, down at the far end, the Florida Panthers, Columbus Blue Jackets, and Winnipeg Jets. So there's a huge spread in between these 2 lines. So then we're trying to now interpret that into the amount of money that investors will have to include into an NHL expansion team, so that the team doesn't have to be as successful as the Boston Bruins, but so it can get chronically infected fans that purchase merchandise. And so I'm going to stop there, and hopefully you have some questions for me. I can take questions about anything that I've talked about, and if you also have questions about certain diseases that you've heard about and how they spread, I'm happy to take those questions as well.
Thank you so much. If you can hit the stop share button, then we'll be able to see. Perfect. And I guess the first question is, when we talk about applying this to the spread of disease, you mentioned a couple things quickly about what we do with that information. Could you elaborate maybe a little more on how we apply this knowledge and why it's useful?
Okay, so as part of the Center for Disease Modeling, we have some collaborations with the public health institutes as well, by province and in Canada, and then also with some parts of the World Health Organization and the Center for Disease Control in the United States. And so what we do is that we take a real world question about a specific disease of interest. We try to model it without too much detail, because then we can't get any new results that are informative, because there's too much uncertainty if you put too much detail into a model. But hopefully we have an optimal level of detail that we need. And then we can determine some mathematical results that then we can interpret back to the real world question about that disease. And then we share our knowledge, our new knowledge, to the public health agencies and to pharmaceutical companies, and to the World Health Organization, and to even physicians if it's a disease that a physician is interested in. And then we see if our new knowledge can help change public health policy or how medicine is provided to people.
Thank you. And we have our first question from Hamilton, Ontario for the day, from Lawfield and the grade 7s there. They would like to know, can you tell us how this relates back to vampires? I guess we haven't delivered on that promise yet.
So what we were interested in was trying to model how fast the vampire population would actually be able to spread compared to zombies. And we see that vampires actually can persist in a population in movies. So vampires and humans can coexist at a nice equilibrium. So what we tried to do in this book that my colleague and I have published this work in is try to see if we can find an infectious disease that we can use to model vampires and find one that we can use to model zombies. So we likened zombies to the plague, and we likened vampires to the spread of HIV, because people who have HIV can live for a very long time, and we have this coexistence between people who don't have HIV and people who do. So we just tried to get an idea of model structure, and we found out that vampires were happy to live with them.
There you go. So now we've definitely talked about vampires, we've talked about the NHL, we've talked about zombies, we talked about actual diseases. I think we've got our bases covered for what we promised to talk about. And now a question from Ms. Falconer's class at Lakeshore. She says they're currently working a lot with graphs and charts, and it looks like you use a lot of these in your work. Is that true?
Yes, I am always using graphs and charts. Some things that I do where I take data, I like to be able to see the data that I'm working with. So I'm making histograms and scatter plots to have to see if there's relationships between different data points. I'm also always using a slope. So if you have one point on a line and another point on a line, then the difference between the height of the 2 points and the width between the 2 points is the slope. So it's delta y over delta x. And essentially, almost all of my work is based on what the slope is on a line at a particular point. So I'm always using graphs.
I have to say I'm not surprised. Graphs and charts infiltrate a lot of research, not just mathematical modeling.
Yeah.
So next we go over to Braeside School, and Yon wants to know how the flu spreads. And Jillian from the same class wants to know how many people have died from the flu and how that happens.
Okay. So the flu spreads, the flu is a respiratory infection, which means that it comes in through your respiratory tract, so through breathing and your mucosa, through touching your eyes if you've touched something that's infected. And it goes down into your upper respiratory tract, and it starts to infect cells that are part of that part of your body. And then that really means that now that part of your body is making more mucus, because mucus is being made so that the mucus can help you cough out something or sneeze out the disease. That's your immune system trying to get rid of something. But also, as the mucus is made, it makes it harder for people to breathe, and they get stuffed up, things like that. So how the flu spreads between individuals is if someone is coughing, or maybe they're not sneezing into their elbow, they're not being careful with how they're spreading, then if you're sitting beside someone who has the flu, you might be able to get it. Also, the flu can live, the pathogen can live on surfaces for a while. And so if someone who's had the flu has sat on this seat of the bus, and they've contaminated that seat, and then you go and sit on it a little while later, and you touch the seat, and then you rub your eyes, then you might be able to get the flu as well. So moving on to the second part of the question about how people die from the flu, well, that may have something to do with their immune system not being very effective in getting rid of the disease. And maybe they also have some other conditions that are affecting how well that they can deal with the disease. So if someone has a condition that affects how they can breathe, then having an infection in your respiratory tract is going to also affect that. And then you can think that also the pathogen is not really just only in your respiratory tract. Some pathogens can get into your blood. And then maybe there will be some other super infection of another disease that will be taking advantage of the fact that someone's already sick. And then someone can also die because they're just so sick that their immune system can't deal with getting rid of anything.
Excellent. I'm working here at keeping up with these questions. It's great. We've got lots coming in. Moving on from the flu, can we move back to talking zombies? We've got Justice, a grade 6 student at Edgemont School, who asked, would the zombie chart change if there were extra variables that changed, such as who the people were that were survived or why?
Yes. So we're hoping that we'd be able to survive, at least some of the humans would be able to survive longer than a month, or maybe some of the people wouldn't actually be infected. And so that chart would change if, for example, we moved people to an island. Zombies so far we've seen in movies don't swim, or at least I haven't seen zombies that swim. And so if people on islands maybe are more protected from that. Or maybe there's a vaccine, like so in the movie 28 Days Later, they developed a vaccine. And if we can give that to people, and if it's an effective vaccine, then some people will not have to be infected. And then we've also seen in some other movies like Shaun of the Dead, where the zombie plague actually just kind of went away after a while. And then at the end of the movie, they're coexisting with some of the zombies too. So the idea is to put in control strategies there that can prevent the disease from infecting everyone and also moving it further along.
Right. And moving on here to a question from Ms. Kang's class. Erica wants to know where the flu starts each year. And maybe in general, we're talking about the spread of these diseases. How does it start?
Okay, so the world is globally connected. So there's lots of spread of diseases through airplanes and transportation. Also, though, there are some pathogens that can persist all year round in the population. So we kind of think of the flu as having a flu season, but there are some people who can get the flu in the summertime too. So maybe the flu is really just persisting in our population, and we know that the optimal temperature and humidity level in the air for the flu to spread is during the flu season. It's not very good for it in the summertime. So we would see less flu then. So if it's persisting in the population, then we would see less flu in these summertime and more in the winter. And then we have people that are traveling all over the world, and we can spread the disease that way. So the flu, though, when we're trying to determine what the vaccine will be, we have to make that decision about 6 months ahead of time, ahead of our flu season, because it takes 6 months for the vaccine to be made. And so really, the people who decide what should be in the vaccine have to make a prediction of what the flu strain will be 6 months from now. And so they look at what's happening in the southern hemisphere to try to figure out what vaccine you'd need in the northern hemisphere. And then they will look at the northern hemisphere circulation of the flu to see what vaccine needs to be made for the southern hemisphere time of year.
Right. Lots of different researchers collaborating, it sounds like.
Yes.
So we talk a lot about the spread of disease and mathematical modeling. How would you contain the spread of the Ebola virus based on this information? Are there other variable or test groups? This question comes from Ms. Dubois' class.
Okay, well, here's a nice Canadian story. There's a vaccine that is being made that's being tested against Ebola, and that was made in Winnipeg. So shout out to Winnipeg and the NRC. So that's something that is a great invention for Canadians. But also, if there is no vaccine, what can we do? Well, we know that individuals that have died from Ebola can still transmit it after they have died. So one thing that was practiced during the more recent Ebola pandemic was making sure that individuals who have died from the disease are handled in a better way. So changing the way that some funeral practices have been done in the past so that people who do not have Ebola and who are susceptible won't actually contaminate themselves from handling the bodies. Also, just being sure on how to social distance yourself. So if someone is sick, you can help them, but make sure that you're wearing proper coverings so that you're not going to contract the disease. Or if you touch someone, then you wash your hands, things like that. And that will change the parameters in the mathematical model, and we'll be able to see how effective these different interventions are.
Excellent. So you could predict that and then know which advice is the most important to give people to prevent the spread. Is that right?
Yes. We can test a vaccine, a drug, social distancing, and we can see how immediate the effect will be on it for different control strategies.
Right. Okay. And from Sonalta School, they'd like to know if you can tell which countries or what parts of the world are more likely to break out in a life-threatening disease.
So places that are more likely to have a life-threatening disease. So there are different variables that you need to consider there. So first, where there is a large population density. So if people are living very close to each other, then it's easier for a disease to be transmitted. In places of the world, there aren't as good healthcare resources as we have. So places that have high population density and don't have very good healthcare resources are more susceptible to having these bad things go through their populations. And then it also depends on where you live in the world. So if you live in a temperate zone or a rainforest or a desert or things like that, all of those matter because the temperature in the air and the humidity level will also determine what pathogens can reside. And then you can think about then too what wildlife are living around the area and what insects. So we know mosquitoes can carry some diseases, but different types of mosquitoes carry different diseases. So what mosquitoes are prevalent in that area, that tells us whether something like West Nile virus would be there or if something like chikungunya will be there, things like that.
Excellent. Now we're just about out of time, and Ms. Kang's class asked the last question I was already going to ask, so great minds, students there, we think alike. What would the career path look like for students who might be interested in this field of work in math? Okay, so certainly making sure you go and do all those math courses in elementary school and high school. Going off to university and either majoring in biology. We have a degree program now at York called mathematical biology, majoring in applied mathematics, and then some connection also to computer science. There are people who are educated in engineering or in physics as well that are mathematical biologists. So something like that, going off into an academic stream. And then people who are in this field that maybe aren't at universities, but work for public health agencies and hospitals. There are lots of statisticians and mathematicians that also help out all of these other places. And if you're interested in learning more about mathematical biology and disease modeling, I'm always happy to, well, York has a Science Speakers Bureau, so I go out and I visit schools in the GTA and up into Simcoe, Muskoka, and in the Peel region. So if you're in that area, I can come out. And if you're not in that area, then I urge you to talk to your local university to see if you can have someone come out to your school. And my network, the Center for Disease Modeling, is across Canada. So we probably do have someone close by. Excellent. Well, good to know. We have some more resources to go on beyond just today. But thank you so much, Dr. Heffernan, for joining us and answering our questions today. That was wonderful. Thank you so much for having me, and hopefully I can have more people from the CDM do these PIR
live events as well. That would be great.
Anytime. So next week on PIR Live Event, we're talking about robotics. More information on this and other upcoming PIR Live events is available at www.pirweb.org. Thanks for joining us and have a great day, everyone.